All right. Welcome. Today's essential question is, what are the steps to the different phases of the cell cycle? So a little review on asexual reproduction between prokaryotic cells and eukaryotic cells. On prokaryotes, it does binary fission, where it just simply copies the DNA and then splits off into two identical cells. In eukaryotes, this process is called mitosis, and this happens for any time that your cell tissues need to make new copies of itself in order to repair, replace, or reproduce new cells. So here's where our notes pick up. There are three major stages in the cell cycle. The first stage is called interphase, and there's three different parts to this stage. Within interphase, there is the G1 phase, which is 90% of all cellular life is found in this stage. This is 90% of the cell's life is in the growth stage of interphase. Then it goes on to S phase, and then the G2 phase, which is the second growth phase. And so after it's done growing, it'll go into mitosis, and then the last stage, which is cytokinesis. So here's an overview, a pie chart showing you all the different phases of cell division. So here's that large stage interphase where the cell spends most of its life in. And then here, just for a quick bit of its life cycle, is in mitosis. And then the very end, cytokinesis, which is the end of mitosis. So let's take a look at interphase, that first stage. This G1 phase of interphase, the cells do most of their growing during this phase, and they make new proteins and organelles during this stage. So it's the major growth cycle of the cell. Then an S phase, and it's called S phase for a reason. It stands for synthesis. So the cell is making more DNA replication of chromosomes. So this is when the DNA is being copied, being replicated for cell division. And then it'll go into that G2 phase, which is the second growth phase. The organelles that are needed for cell division are produced during this stage. And a couple organelles that are made are the spindle fibers, which will attach to the centromeres. Remember what those are from chromosomes for division. And then the centrioles are also made, which attaches the spindle fibers. So the cell is getting ready for division. It's making the organelles needed for division. During the S phase, this is when the chromatids are replicated and made into two copies and made into two sister chromatids. And so remember, they are attached together at this centromere. And so then getting on to the next stage, mitosis, this is when the nucleus divides into two nucleus with the same number of chromosomes. So one new cell has the same number of chromosomes as the other cell, and then the other new cell has the same kind of chromosomes as the original cell. So both cells have the same number of chromosomes and the same kind of chromosomes. You're making an exact copy of the original cell. So here's kind of just a quick overview of what mitosis looks like. And we'll go over all these different phases of mitosis, but as you can see, the chromatids are replicated and made into chromosomes with the two sister chromatids attached at the centromere. They line up, split apart, and then each daughter cell receives the same number of chromosomes and the same type of chromosomes. All right, so we're going to go step by step through the different steps of mitosis. And so if you can remember this, these four letters, P-M-A-T, PMAT, that will help you memorize the order of mitosis. So the first phase, the P phase, is prophase. And so I like to think of prophase as pieces. Pieces such as the nuclear envelope breaks down. Spindle fibers start to form, and then the chromosomes coil and condense, and the duplicated DNA becomes visible. So this is the pieces or prophase stage. This is when all the pieces start to come together for cell division. So let's take a second and pause the video. I want you to get a drawing of prophase, a cell that's in prophase. I want you to, there is a checklist in your notes for all the different pieces to draw in this stage of the cell cycle. Then the next stage, metaphase, is I like to call the middle phase, because the spindle fibers start to attach to the chromosomes, and the chromosomes move to the center of the cell. So they start to line up at the center of the cell, or the middle of the cell, because they eventually are going to split off into two cells. So again, take a second and pause. There's a checklist for what to include in your drawing, and draw a picture of a cell in metaphase. Then it goes into anaphase, and I call this the away phase, because the two chromatids separate, and now they're called chromosomes, and the chromosomes move to opposite sides of the poles of the cells. So they are splitting and going away from each other, and they're about to go into two separate cells. Again, pause and draw a cell in the stage of anaphase. Then the very last stage of the PMAT of mitosis is telophase, or 2. I call it 2 because this is when the division really starts to occur and the nuclear envelope starts to reform and the chromosomes uncoil, and so it's really starting to form into two separate cells. You start to see the nucleus coming back together, and you can see that these chromosomes are on separate sides of the cell, and you will start to see these microtubules and all these other different pieces starting to disappear, because the cell is almost done with division. So take a second, pause, draw a cell in the telophase step. Okay. Then the last stage of the whole cell cycle is called cytokinesis, and it means the division of the cytoplasm. This is when the cytoplasm divides, forming two separate cells, and the chromosomes decondense, meaning that they're not going to be in chromosomes anymore. They're going to uncoil because cell division is complete. It no longer needs to be in an organized chromosome. So I want you guys to draw cytokinesis because you can really definitely start to see these two cells being split apart and pinched off. Okay, so in plant cells and animal cells, this stage of cytokinesis looks a little different between plant and animal cells. So in animal cells, there's this structure down here that forms, and it's called the cleavage furrow. And it forms as the cell membrane grows together to complete division. So this little section right here between the two cells that are almost completely divided, it's called the cleavage furrow. And then plants have these vesicles that fuse together to form a cell plate. Okay, so here's a cell plate as the new cell wall grows on either side. So I want you guys to draw the two different types of cytokinesis in animal cells, plant cells, and label the cleavage furrow and the cell plate. I should also mention this is further down in your notes too. Okay, then going back, overview of mitosis. Mitosis and cytokinesis produces new cells that are identical to the original cell. It allows the cell to let the organisms grow, or allows organisms to grow. Mitosis allows organisms to replace damaged tissue. And mitosis allows for some organisms to reproduce asexually. So you can see down here, here are my examples. When an organism needs to grow, it needs to produce more cells for more surface area. So mitosis helps with that. For damaged tissue, like cuts and burns, mitosis allows for organisms to repair that damaged tissue. And then for some organisms, mitosis allows it to reproduce asexually, like the starfish here. If the starfish loses a limb, mitosis can regenerate a new limb for the starfish. Okay, so the questions to answer on Google form If you were to observe many cells underneath the microscope, which stage in the entire cell cycle do you think you'd see most of the cells in, and why? So going back to the whole cell cycle, in which stage do cells spend most of their life in? And then question 2, which stage of the cell cycle is this cell in? So this is a cell, or I guess it's almost 2 cells because it's in mitosis. Which cell cycle is this cell in, and how do you know? And I've showed you a label here. These little pieces right here are the chromosomes. All right. I will see you on Monday. Have a good weekend.